Hi, everybody. My name is Dr. Joseph Geraci. I am a professor of molecular medicine at Queen's University in Ontario, Canada, and a professor of medicine at Augusta University in Georgia, United States. I have been straddling mathematics, computer science, and medicine for a decade or more. And I'm also the CEO of Netramark Corp. And within Netramark, what we've done is we've created a technology that does straddle these areas. And one of our main products allows medical scientists to understand the precise ways that complex diseases can manifest themselves. And so this is why I'm excited to introduce this challenge to the Kaggle community from a machine learning perspective. Now, ALS is a complex disease, and we're very excited about the data we've been able to accumulate so far. The first caveat is that this data is not very large. So this is not a deep neural network type of exercise. You still have to create models and you still have to validate them. But what we want from you is a story. And so I'm going to tell you how to potentially go about this. I'm going to introduce you to a cool tool that allows you to interact with the biology, so that as you start to do your feature engineering, feature selection, feature reduction, whatever you want to do, that you can actually explore how it relates to what is known and potentially discover what's not known. Talk about biology and ALS. Okay, so the first thing you want to do is you want to go to everythingals.org and go to the video section and review Dr. Matthew Harm's video, which reviews the genomics of ALS and does a beautiful job. And then if you want to leap ahead, Dr. Bob Brown does a beautiful job at showing us what's being done in this field. I've been very lucky to be able to work with Dr. Leslie Thompson at UCI and Dr. Finkbeiner at the Gladstone Institute. They've helped us assemble this data, and they've been working with me to try to make our own way forward in creating a treatment for this horrible disease. So let's talk about the disease, okay? So the basic way that we view biology is that we have this encoded information about our molecular machinery, proteins in us, in the form of DNA. The DNA gets translated into RNA, into a number of RNA, and that RNA then gets translated into proteins. And a few of the data sets we're sharing with you is actually at that RNA level. We actually give you data about the counts of all the different RNA that we found in these ALS patients. And so what's happening in ALS? So what happens in ALS is that the motor neurons in these patients die. We have these motor neurons in our brain and in our body. And what happens is something goes wrong. An example, if you would like to read about it, is one way of looking at it potentially is that apoptosis is running off. And apoptosis is something that keeps us healthy. It's very important in cancer, for example. And what it is if a cell has an error in it, or mitochondrial damage or something, right? A successful strategy is to tell the cell to commit suicide. And that way it doesn't spread that error to its daughters. And so is it possible that something like this is running out of control? And so this is what we're trying to figure out. Maybe there's a way to stop this, because what happens is either the neuron dies or the actual part of the neuron that touches the muscle through the neurotransmitter starts to retract. The muscle doesn't get innervated and wastes away. And it's painful. And so that's what happens. Now, why it happens, we don't know, but there is something to go with. So when you watch this video by Dr. Harms, I want you to pay special attention to this diagram here. These are a set of genes that the community believes has something to do with ALS, and there might be a key here. However, it might not be the true fundamental key. Maybe you'll discover that. But the reason you want to keep track of this, I'm going to show you why. Okay, so remember KIF5 alpha, just because I picked it at random. And let's see what happens. So this is one of the tools that one of my companies created to help medical scientists explore this. And let's just take a look at what it thinks is going on in one small subtype of 20 people. So what's separate, the AI is very cool here. The AI separated these 20 people because they all have a type of ALS that starts in their limbs. And so it kind of saw that it's different from everybody else, including the people where it starts in the bulbar region. And there's different genes. So what you can do is you can grab a gene, let's say NOX1, and you can go to this other tool that I encourage you to start playing with. So look, if you're a pro at understanding systems biology, protein interaction networks, I encourage you to look at Dr. Igor Gerasica's Navigator tool. It's amazing. You download it, create this, create these maps and these interaction networks. But I'm going to show you how even somebody without experience can do this. So you take a gene, I throw it in here. Let's go back and pick another gene. Let's see, type 1. And we go in here and we can ask it to build a network. So while this is cooking, let me explain something to you. I didn't pick one of these earlier ones like this one here I was excited about. The problem is, see this P at the end? It's a pseudogene, right? Read about what that means. So you have to be careful. Not everything that you're going to pull out of this data set is going to be used in this way, but there's going to be genes that you can. So let's look at what happens. So here it is. So what happens is what this is describing is the way the proteins talk to each other in our system, the way they're part of the same pathway potentially, and so forth. And it's all color coded, and you can review it here. And I'm happy to do further sessions on this and to talk about this. Now, remember what I was saying here about KIF5A. Let's see if this thing that we discovered, these two genes that our AI is excited about, is actually, is actually related in some way to something over there. And I'm just playing around. Let's discover something together right now. And so what you see is the machine's creating a story about how KIF5A might be related to these two genes, NOX1. And you can see there's something going on here. If you pull this out, this TECT system is actually doing something with this KIF. It's co-expressed. And there's other pathways. Anyway, lots to explore. But this basically gets us to the point of where we want to be with this data set. We want you to be able to dive in and be able to extract a story about the data. Like I said before, it's not a typical competition. This is more of an explanation, right? We want, explanatory challenge. We want you to come up with models, figure out if there's subtypes, figure out if there's some particular signatures, if there's models that replicate, even with the small data set we're giving you, small in samples, many, many variables. So please be careful. So look, guys, good luck with the challenge. I'm happy to interact with you, and let's do our best to cure ALS, and good luck.